Assalamu alaikum wa rahmatullah. Today we will talk about peptide and protein. Who can answer this question? Why egg become solid after heating? Learning outcome. By the end of this lecture, each student should be able to describe how amino acids are connected with each other to form peptide and protein. Explain the primary, secondary, tertiary, and quaternary structure of proteins. Identify the type of secondary structure. Describe the kind of the forces that stabilize each order of protein structure. Classify the protein according to their shape. Define the term denaturation. Identify the main function of protein. What is the peptide bond? Peptide bond is a type of strong bond that link two amino acids together to form peptide and protein. The formation of peptide bond is a condensation reaction leading to the polymerization of amino acid into peptide and protein. What mean by condensation reaction? Any reaction that involves removal of water molecule is known as condensation reaction. So, in the polymerization, we remove water molecule from two amino acids to form peptide or to form protein. Again, from the definition, in peptide bond formation, we remove water from two amino acids. From the previous lecture, you studied that each amino acid contains amine group and carboxyl group. So we remove water molecule by remove OH from hydroxyl and hydrogen from amine group to form peptide bond. The structure of this peptide bond is covalent bond. It is strong and it's known as amide. So we convert from amine to amide. Amide, that means it is a salt of carboxylic acid. In biochemistry, it's known as peptide bond. Again, from the definition, we said that peptide bond lead to the polymerization of amino acid. So we can form polymer. Polymer is a large molecule contain more than 2 or more than 3 amino acid. How it is formed? By remove water molecule from two adjacent amino acid to form peptide bond. Each polymer or each polypeptide contain N-terminal and C-terminal. C-terminal, it is a carboxylic side or carboxylic group. N-terminal is amine group. So we can add it another amino acids by formation of peptide bond at C-terminal or at N-terminal. Properties of peptide bond. Peptide bonds are planar, rigid, flexible. What mean by rigidity? Rigidity means cannot be hydrolyzed chemically without extreme condition. That means it is stable bond, cannot be break down at usual condition. It is need a lot of heat or a lot of or strong acid or strong base to be hydrolyzed or to be broken. Types of peptides and protein. When there is two amino acids linked together, it is known as dipeptide. When there is 3 amino acids linked together, it is known as tripeptide. Polypeptide means many amino acids linked together. At some point, polypeptide becomes protein when the number of amino acids linked together is more than 50. What is proteins? Proteins are polymers of amino acid linked by peptide bond. So we say if the number of amino acid is more than 50, it's known as protein. So the protein is a large molecule contain amino acid linked together by peptide bond. Level structure of protein or level of protein structure. Protein is folding. Why it is fold or why it is form more than one level structure? To become soluble and to be fitted inside the nucleus. So we said previously that the protein is a large molecule, so it cannot be fitted inside nucleus without folding. That means there is a level of structure of protein. This level is for level primary structure, secondary structure, tertiary structure, quaternary structure. Primary structure. Primary structure of protein is just the sequence of amino acid. So in the primary structure, the bond found is only the peptide bond. So the sequence of amino acid in the protein, the primary structure of protein. Secondary structure. Protein to become soluble and to become functionally active, it should fold. It is fold by two ways, and it is very important. Secondary structure of protein is alpha helix and beta plate sheet. Both depend on hydrogen bond, so the types of bond in secondary structure is hydrogen bond. Hydrogen bond is a non-covalent bond. It is weak bond, so it can be broken easily. It is not like peptide bond that is cannot be broken at usual condition. It is need extreme condition to be broken. In secondary structure, protein is folding by two ways: alpha helix or beta plate sheet. An alpha helix is like helix. Beta plate sheet is like plate. And there is two ways for folding of protein to form secondary structure: alpha helix or beta plate sheet. A bond that stabilizes alpha helix and beta plate sheet is hydrogen bond. So how alpha helix is formed when it is stabilized by hydrogen bond? How beta plate sheet is formed, like plate or like paper, folding paper. Also it is stabilized by hydrogen bond. Third level of protein structure is tertiary structure. Tertiary structure results from the folding of alpha helix and beta plate sheet, where the more folding of secondary structure lead to the formation of tertiary structure. Interactions involved in tertiary structure or bonds that stabilize tertiary structure are hydrophobic, hydrophilic interaction, hydrogen bond, and disulfide linkage. All this bond is involved in stabilize of tertiary structure. Hydrophobic interaction, salt bridge, hydrophilic interaction, and disulfide linkage. All this bond is involved in stabilization of tertiary structure. Here are examples of tertiary structure of protein in which there is a beta plate sheet, alpha helix that more folded to form tertiary structure. Quaternary structure or four-level structure of protein is association of more than one polypeptide. For example, hemoglobin contains 4 polypeptides. Association of these 4 polypeptides form quaternary structure. Each unit in quaternary structure is known as subunit. Subunit or monomers can be identical, homopolymeric, or different, heteropolymeric. The bond that stabilize quaternary structure are hydrophilic interaction and hydrogen bond. So quaternary structure is association, or it is more folding of protein to become functionally active and to become soluble. At the end, for the revision, there is a 4-level structure of protein: primary structure that contain only sequence of amino acid linked by peptide bond. This primary structure is folded to form secondary structure, and it is folded by two ways: alpha helix or beta plate sheet. Association or a more folding of alpha helix and beta plate sheet led to the formation of tertiary structure. Some protein contain more than one subunit, so it is associated or association of more than one subunit to form quaternary structure. In the quaternary structure, in the tertiary structure, protein is functionally active and it is soluble and it can be fitted inside the nucleus. Classification of protein. Protein is classified into two types according to their final shape, after association. Globular protein or fibrous protein. The final shape of a globular protein is spherical, where a fibrous protein is long and thin fiber. Example for globular protein: insulin, hemoglobin, enzymes, antibodies, and myoglobin. Example for a fibrous protein: creatine, elastin, and collagen. Hemoglobin, a final shape after folding, is globular, so it's known as globular protein. Creatine, a final shape after holding, is long or thin fiber, so it's known as fibrous protein. Denaturation. What means by denaturation? Denaturation is a breakdown of all non-covalent bonds in a protein, causing loss of the shape and function, but does not alter its primary structure. In the primary structure, the bond involved is only peptide bond. As we mentioned previously, peptide bond is very strong bond and cannot be broken by unusual condition. Another level structure of protein, secondary, tertiary, quaternary structure, involved or stabilized by a non-covalent bond, and this bond is weak and can be broken. When protein loses this non-covalent bond, become insoluble, become at primary structure or at primary level. So denaturation, that means protein become insoluble, become unfunctional protein. How protein is denatured? Protein is denatured by three ways: physical agent, chemical agent, or biological agent. Physical agent, for example, increased temperature. Chemical agent by change pH, at basic or at acidic pH, or addition of salt, concentrated salt. All these factors cause denaturation of protein or cause lose the function and the function of protein. So it can be denatured by biological agent like bacteria. Why proteins are important? Because protein have many functions inside body. It have structure and support like collagen, transport like hemoglobin, control and regulation some hormones, protection and defense, for example antibodies. Most important function is catalysis. Catalysis or enzymes, its compound or its protein increase the chemical reaction inside body. Now you can answer why egg become insoluble after heating.